Hello everyone, I'm Amrita Singh, Department of Pharmacy of IC College of Engineering and Technology. Today I'm going to present a topic on the pharmacovigilance methods, or different methods to evaluate the vigilance of the drug, the adverse effect of the drug, how the causality, the way of reporting, how they are being registered in the in the format. So as you all know, the aim of the pharmacology is to improve the patient care and the safety of the health, public health and the safety. Also, it contribute the assessment of the benefit, harm, effectiveness and the risk of the medicines. Basically, we can say a risk or benefit method. It also promote some clinical as well as the educational trainings for the vigilance students, and to promote the effective communication to the public so that they will be very vigilant about the areas. Also, it promote the rational and the safe use of the medicines. So as we know that the vigilance part of the drug is being shared, the responsibility is being shared with the different uh, product companies or the pharmacists with the regulatory authorities. So here they are lined up with different responsibility. Like if we talk about the company, they are the legally and morally responsible for the monitoring their product, as well as for the regulatory authorities they're responsible for the safety of the drugs, their licensure, the approval of the licensure. Next is the doctors. They are responsible for the patients who are reporting the area, who are experiencing the areas. Next is the pharmacists and the nurses. Their responsibility is totally focuses on the patients. Let's talk about the different types of methods. So here I have listed around 6 types of methods. First is the passive, active surveillance. Third is the stimulated reporting. Fourth is the comparatively observational studies. In the observational studies, we can be we will be going to read about, check out about the cross-sectional studies, some cross-control or cohort studies. Fifth is the targeted clinical investigations. Lastly is the descriptive studies, which has been implemented on the natural history of the disease or the drug utilization studies. So let's start with the passive surveillance. In the passive surveillance, they are divided into 2 types of reports are come under the passive surveillance. First is the spontaneous report. Second is the case series. So if this active and the passive surveillance goes side by side. So here I have written a difference between active and the passive surveillance. If we say about, we differentiate this active and passive according to the data, then we can say the data which is being actively collected, the patient comes, the health system staff goes to the patient, we are taking part, the healthcare professionals are taking part to gather the data that are known as the active surveillance. And the data, the second is the passive surveillance. Here we are collecting the reports by passively. It means like the data is being itself reported by the health systems, passive form, in a passive form. If we see an example, like for the active surveillance, the health workers goes house to house and detect the fever cases or the areas. In that, they collect, for the confirmation, they collect the blood slides or the blood samples. These are come under the active surveillance. If the patients are coming to the practitioners, healthcare professionals, public healthcare professionals, or the community healthcare commissioners, private practitioners, then it is a passive surveillance. So mostly used when diseases is targeted for the elimination or the reduction. When these are being targeted, this is an active surveillance. When the disease is targeted for the control form, that is a passive surveillance. In the active and the passive surveillance of the organizations are been included, like it includes active includes NBDCP, NLEP, and next passive surveillance, they are work under the national healthcare programs, which is being provided by the India. And the active surveillance are most expensive than the passive. If we see about the diseases, an example like disease like poliomyelitis, leprosy, neonatal tetanus, these are the examples of the active surveillance. And next is the passive surveillance. The examples are diphtheria, hepatitis B, or the mumps. These are the some surveillance examples. Let's talk about individually. This first is the passive surveillance. As I've told you, these are actively participated, and the healthcare professionals are going to the patients to collect the data. So the routine reporting of the health data, it should be, first of all, should be a notifiable disease, as well as with the risk factors, all the health related events which is being experienced by the patients. Okay, so next, the healthcare practitioners report the events as they come to their attention, and they this type of surveillance is more common type, because the report is being reported by the healthcare professionals, which will be genuine and being assessed by the patient, healthcare professionals. Let's see the example. The physician sees a patient, diagnosed the measles, and then initiated a case report by contacting the local healthcare department and providing the details as required for a case of the measles. So it is not been a huge burden for the reporters who is the patient who is suffering. Next is the spontaneous reports. In this, the voluntary reporting is being done by the patients. As the patient is being admitted in the hospitals or the outpatients, they come to the, they will inform the healthcare professionals about the experience of the adverse drug reactions. So all the voluntary communication by the healthcare professionals or the consumers to a company, then a regulatory authority or other organizations will take part in the spontaneous reporting. So it defines one or more adverse effect in the patient who was given one or more medicinal products, or some identification of the safety signals which is being which is being launched in the market, and it is being used in a for a once. And also can vigilant a company to read adverse events that were being notified or noticed in earlier clinical trials or other than the pre-marketing studies. Next class of this and the spontaneous reportings deliver important information about the risk groups, who which groups are more at the risk to produce the ADRs. So in this active surveillance, we will find this major goal. Next classification of active surveillance is the case series. So next, reporting all the series of case reports which deliver the sign of an association between the medicines and an adverse events. These are the case series, and these are very valuable, and it can be saved for a further evaluation of the ADR. Next is the stimulated reporting. Stimulated reporting come under your this is the third part, which is after the active surveillance. We have seen the passive surveillance. In the passive surveillance, we have seen spontaneous reporting or the case series. Next is the stimulated reporting. Stimulated reporting, in which any of the new if any of the new product is being launched by the company will contact to the healthcare workers and provide a safety information about the new product. They will give all the necessary information about the new product, about these any event which is being seen during the clinical trials. Also ask the healthcare professional to submit the spontaneous report if after giving this medicines, the patient experienced any of the areas. So this report is regarded as a spontaneous event reporting. This method is being encouraged and facilitated by the healthcare professional for all the new products or for the limited time period. Here we can collect a lot of information during one go. And these methods can be also online reporting of adverse event during a spontaneous reporting. So this is one of the major used uh, reporting system, but the drawback is the data are more often incomplete because some companies did not provide the full information. Some information is being lacking behind the risk. So the data are often incomplete. Due to that, if we are not able to produce a accurate incidence rate, we can just make a hypothesis report, not the particular accurate data. Third is the active surveillance as the opposite of the passive surveillance. Here, the reporting of the disease, the risk factors, or the health events is being initiated by the patients, and the patient is report the areas. So here we have when there is an indication that something unusual is occurring, the patient comes and report the ADR, determining the particular number of adverse events through a constant pre-organized process for informing or confirming the adverse events. So in this, there are different set of methods. Like first is the senatorial methods, which is a highly quality data which required a particular disease, in which they like according to the community or the geographical area is being selected, which is rich in the informations from they select that area and report all the units with a high probability of seeing the disease in the questions. They prepare a questionnaires and they ask a particular geographical area about the events, and also good laboratory facilities is been provided, and experienced well qualified staff is very important to identify and also notify the certain diseases is disease or the ADR is being reported by that community or the geographical area persons. In this, we have the set of process like making a record, revising the record, or questionnaires, whatever been prepared by the patients or the physicians during the section of during the senator sites, whatever the site they have chosen. So they are being modified time to time according to the diseases. Next, the major weakness of this limitations of this site senatorial site method is like it is difficult with for the selection bias. Uh, we cannot do a selections of the numbers of the patient. Due to that, a small number of patients we get, and the augmented cost, the cost is more and the patient is less, so the data will not be accurate. Next class of the active surveillance is the drug event monitoring, which is very common and important also. So in this drug environment monitoring, the process we are using is the cohort based study or the prospective or the observational.